Welcome to Science Daily. In this episode, we learn about genetic sequencing, from the very basics of what DNA is to complex issues such as handling all that genetic information. Enjoy. The fascination with DNA all started 60 years ago when famed scientists Watson and Crick found out DNA's shape, double helix. Once it was understood how it fit together, the next step was to find out what it meant. But in order to do that, we needed to have a firm understanding of what DNA is. So let's take a look. The entire DNA molecule is a polymer made up of small building blocks called nucleotides. These nucleotides in turn are made up of 3 smaller building blocks, 1 phosphate group, 1 sugar, and 1 nucleobase. To make our lives easier, there are only 4 of these bases in DNA: adenine, thymine, guanine, and cytosine. And the arrangement of these bases throughout the entire molecule are what make up your genetic code. Each specific region on the ladder does certain things, so one region might tell the cell to make a specific protein, while another has the blueprint for the protein in question. Each of these regions is called a gene, and all the genes in DNA are collectively referred to as the genome. Since we're all humans and relatively similar, our genomes are pretty similar too. In fact, if you take 2 average people and compare their entire genome, that is, the exact order of all 3 billion nucleotides, you'd find that they're over 99.9% identical. Only 1 in every 1,000 nucleotides is different, and somewhere in that 0.1% is what makes everyone unique. Their eye color, the curliness of their hair, right down to how likely it is that they'll have certain kinds of cancer. Looking at and understanding these differences is exactly what the field of genetics is about. Instrumental to genetics is sequencing, the act of taking DNA, running it through a machine, and finding out the exact order of all 3 billion nucleotides. In this technological age, no corner of the tech industry is immune to improvement, and since sequencing is done by machines, they too are participants. Except that instead of just getting faster, genetic sequencing is getting cheaper. Which raises the question, what does that mean? Our program today, in 2 parts: what's driving the decrease in cost of sequencing? And what does this mean for your everyday Joe? Please stick with us. Part 1 of our program today, the new machine on the block. At the University of Washington Medical Center's main hospital, scientists are excited because very recently, in fact, just about a month ago, they received the cutting edge in genetic sequencing technology, HiSeq 2500. This new machine can test over 3 billion single nucleotides at once, meaning they can perform the Oncoplex V2 panel, a genetic test that looks at over 100 different cancer-related genes in order to find the best way to treat a patient's cancer. But in order to understand why these scientists are so excited, we need to go all the way back to 1974 and a little-known paper published by Dr. Sanger describing his method for sequencing, the Sanger method. Dr. Sanger's method for sequencing goes by a variety of names, from as simple as Sanger sequencing to chain termination or dideoxy sequencing. Whatever people call it, Dr. Sanger's method is the basis for all current and next-generation sequencing methods. Dr. Sanger's method relies on a unique form of molecule, which is nearly identical to the nucleotides we talked about earlier. These new ones are called dideoxynucleotides, or DDNTPs for short. Just like with the earlier nucleotides, they have a phosphate group, a sugar, and a nucleobase. In fact, the only difference between DDNTPs and the nucleotides I already mentioned is the addition of a hydrogen group on the 3' carbon instead of a normal hydroxyl group. This minute difference of only 1 oxygen is the basis of the entire method. These DDNTPs can only bond to 1 nucleotide, meaning that they end each DNA strand and can't continue onwards. So let's say you have a DNA chain 10 nucleotides long, and you want to find out exactly what the order of the nucleotides in it are. Well, if you could get a version of the strand for each possible length, so 1 strand of only 1 nucleotide, a second strand of 2 nucleotides, a third of 3, and so on, since each of these strands is 1 nucleotide different in length, it becomes easy to compare them with a process called gel electrophoresis. Gel electrophoresis basically uses electrical charges to push a molecule through a gel substrate. The smaller the molecule is, the farther it travels through the membrane. So naturally, if you wanted to sort out all the small strands of nucleotides you have, you could run it through gel electrophoresis, and you could then see which ones traveled farthest in the gel, and you know that those are the shortest ones. Dr. Sanger realized that since DDNTPs only bond to 1 nucleotide, that they would naturally come at the end of strands of DNA, and that he could use gel electrophoresis to sort them all by size at the end. The key discovery is that when DNA is replicated, it uses normal nucleotides and ddNTPs at random. So if you put a bunch of ddNTPs along with normal nucleotides and performed a whole bunch of DNA replication, like in polymerase chain reaction, then you could get every imaginable chain length possible, and all of them would have the specific ddNTP at the end. So Dr. Sanger described a method where you could put one type of ddNTP in a vial, ddCTP, the one that replaces normal cytosine nucleotide, along with all the other components for DNA replication, DNA polymerase, lots of the 4 other unaltered nucleotides, and DNA primers in a C tube, and then replication occur. After the DNA replication is done, what you end up with is a bunch of strands that end at every cytosine nucleotide in the DNA strand. So some strands start at the beginning and end at the cytosine that's 3 nucleotides from the start, while other strands begin at the start and end at the cytosine that's 100 nucleotides later. The great thing is that you can get all the strands in between as well. Still following? Because Dr. Sanger certainly was. He realized that if he did the same exact thing for the other ddNTPs, the ones for adenine, the one for thymine, and the one for guanine, that he would end up with strands of all the different possible lengths of the original DNA strand. So when he runs each reaction tube, each in its own section of the gel electrophoresis machine, he could literally read the sequence of nucleotides by looking at which strand went farthest, noting which of the 4 reaction tubes it came from, and then looking at the second farthest traveled band and seeing which of the band it came from. In this manner, Sanger described a process to actually determine what the order of nucleotides in DNA was. I know, it's difficult to understand, so let's do a quick summary. Basically, the Sanger method uses 4 different reaction tubes, one labeled A, one C, one T, and one G, one for each of the different nucleotides. Into these tubes are placed a special nucleotide that only can bind on one side of it. Because these special nucleotides can only attach on one side, they always end strands whenever they're attached. So what you end up with is at least one strand of every single possible length you could get for the gene you're sequencing. And now that you have all these lengths, you can just run them through a gel electrophoresis machine and look at the result. By looking at the results, you can tell what order they're in. And then all you have to do is look at what reaction mix they were in, and you can see what nucleobase it is too. So basically, you can actually get a genetic sequence just by looking at which base is where, and then which one follows that. It's as simple as that. This method was described in 1977. Since then, many advances have been made. One of the first advances to be made was developing a way to radioactively mark all the ddNTPs so you could do the gel run all in one well instead of having to waste 4 wells. The next step for scientists was to get computers that could read these results for them so they didn't have to waste their time interpreting the results. So instead of going through by hand and writing down the sequences, they were instead given a file by the computer that had all the sequences for them, saving a lot of time. This computerization of the recording of results has led to much faster turnaround times on genetic sequencing. But the biggest impactor on turnaround time to genetic sequencing is the ability to sequence more information at once. With Sanger sequencing, only relatively short regions can be sequenced, no more than 100 nucleotides long. Through revisions and improvements to Sanger's methods, they were able to increase that up to 1,000 nucleotides long. But even that's pretty small. I mean, just think about it. The human genome is 3 billion nucleotides long. So really, it pales in comparison. In order to solve that, scientists have made new ways, such as shotgun sequencing, that break that larger DNA piece up into fragments and then sequence those. They then feed all those small sequences into a computer where it assembles them all together based on overlapping regions in order to produce one large sequence. Shotgun sequencing can sequence genetic pieces as large as human chromosomes, but even then scientists aren't satisfied. Research is being done around the globe, at Harvard, at Oxford, and even right here in Seattle at the University of Washington, all in an effort to make it easier to sequence larger portions of DNA. As a result of it being easier to run larger pieces of DNA, the cost of sequencing is going down. In fact, it won't be long until it takes less than $1,000 to sequence an entire human genome. But once you have the genome sequenced, the real trouble is finding out what it all means. And for that, we go to part 2.